because everything's built on cells. Then we get into the system in general. So we'll be going through the spinal cord. So test 4 is going to be the muscle system, nervous system, spinal cord. Then on test 5, we'll do cranial nerves and the brain. We'll do SNS and the ANS. So it's one of those that we'll be building. Obviously, there's going to be a lot of crossover. Some of the structures and functionality we see with the spinal cord, we're also going to see with the brain. So once we learn it on test 4, it is something that might help us with the test 5. So there's going to be a little bit of double dipping, making it a little different than all of our other tests where everything was kind of its own isolated sphere where they didn't cross-contaminate each other. So we're going to be using the information that we learned in this chapter on this test, but obviously on test 5 because we're still looking at nervous tissue. So in theory, test 5 might be a little easier because we've experienced some of that content on test 4. So it'll be a little bit of a repeat when we get to it in test 5, just at a different angle. Oh, we looked at the spinal cord and how it relates to the spinal cord on test 4. On test 5, we looked at the thing and how it relates to the brain. Okay, make sense? So when we look at functionality of the nervous system, it's going to allow us to control our body, when to sweat, when to move, whatever. Helps with communication within the body, communication from outside sources to inside sources, vice versa. It's going to help us control homeostasis. So way back on chapter 1, when we looked at homeostasis, we had 2 major systems that do that. We've got the nervous system that gives us our day-to-day functionality. You know, if your hand is on fire, you got to tell it to move. And then we have the endocrine system, which is going to use hormones for long-term things like ovulating and growing and things like that. So that's more of your AP2 stuff. All the different functionalities, all the different things that the nervous system does for us can be categorized in 3 different categories, 3 different types of functions. So our first one is monitoring. This is sensory input. Whether we pick up that it's cold in here and we tell our brain, so we might shiver or we might put our coat on, or we tell our brain that our stomach is full from the stomach being stretched out too much. Anytime we have sensory input coming in, that's going to be a big job for the nervous system to do. So it's going to pick up the stimuli in the variable, whether it's external stimuli or internal stimuli. Integration. Once we get that sensory information, we need to decide what to do with it. Do we need to respond? My stomach's full, I should stop eating. It's cold, I should start shivering. Maybe I should put my coat on. We need to take that sensory information and process it and decide a response that we need to have. And then finally, the motor output, the response. So if it's cold in here, the brain might say, okay, we need to get goosebumps. We need to start shivering. We need to stop sweating. It might tell you to put a coat on. If your stomach's full, it's going to say stop eating. So you feel full, you're not going to eat anymore. So you're going to have that motor output. The effector organ is going to carry out whatever command it was given by the integration, the processing centers, so we can carry out. So we can pick up information, process the information, and then respond to that information. When we look at the divisions of the nervous system, so we have our first division between the central nervous system and the peripheral nervous system. So the CNS and the PNS. When we look at these, again, there's a lot of slang in the nervous tissue. So it's one of those things that just to make you feel that much more in awe of brain surgeons, they have their own set of language and words that you have to learn to understand what those crazy people are saying. So unfortunately, you have to learn what those crazy people are saying. That's half of the job of a nurse, is to translate doctor speak into something that patients can understand. And with the nervous tissue, there's a lot of language. There's a lot of new words, definitions, and so forth. So there's some of them. Central nervous system, the CNS, that's going to be the brain and the spinal cord. Those are the only 2 components of the central nervous system. They're going to have the job of integration, of thinking. So they're going to take that sensory information, decide what we need to do, and then they're going to tell the body how to proceed. What should they do in response to that stimulus coming in? Now, the responses are based on reflexes, so something you might have developed over time. So if you play catch with somebody over and over and over again, eventually you throw a ball at them, they're going to catch. Okay, it becomes second nature, it becomes a reflex. They don't have to think about it too much. Experience. What you've experienced in the past can alter how you're going to respond to that new experience coming in. So no matter how many times you try to play baseball, you just can't catch the ball. You might duck when the ball gets thrown at you instead of catching it. Okay, so you develop a different type of response because of the experience that you've had in the past. And of course, current conditions. What's going on right now and how is that coloring your judgment? And this is something we've all been aware of, right? Your past experience makes you who you are and how you respond to what's going on around you. So if I was to say 70 degrees, okay, if it was 70 degrees tomorrow, what would we consider that based on the past few days? Warm, right? Oh, there's a hot front coming in. It's going to get hot. It's going to get warm. We went from 10 to 70, okay? Heat wave. Now, if I said 70 degrees in August, how would you perceive that 70 degrees during August? Cold. It'd be a cold front, right? Because August is 900 degrees. Okay, so it's a cold front whenever you get 70 degrees. So in August, 70 degree comes in. You might put on a light jacket because it's a little chilly out there. But 70 degrees rolls in the middle of January or February. My gosh, it's a heat wave. You know, take your jacket off. Relax, be comfortable. So even though the temperature didn't change, it's still 70 degrees. What we've been experiencing, what those conditions are going to alter how we perceive it, even though the thing that we are perceiving hasn't changed at all. Peripheral nervous system, everything not the brain and spinal cord. So cranial nerves and spinal nerves and the nerves in your hands and all kinds of stuff, that's not brain, not spinal cord. They're going to do the other two functions, the monitoring function and the motor output function. When we further divide, now some books will further divide the central nervous system into brain and spinal cord, so you know there's a distinction between those. I don't really look at it that way because they both have the same job. So why divide it up? They're doing the same thing. So sometimes you will see flow charts of the divisions of the nervous system and they'll divide the brain and spinal cord. Other flow charts won't. This depends on your book. Now with the peripheral nervous system, we have two functions there. So that means we're going to divide the peripheral nervous system into two. We're going to have the sensory guys and we're going to have the motor guys. Okay, so that's our first logical division there. So the sensory division, also known as the afferent system, because afferent means towards. We learned these terms back again when we did homeostasis on test 1. So afferent always means towards. So sending information to the brain, to the spinal cord. So sensory input coming in. Then we have the efferent system, sometimes referred to as the motor system or the motor division, because this is the input that has been processed by the brain and spinal cord and going to the body's organs, the effector organs, so you can start sweating if you need to sweat, or start shivering if you need to shiver, or release whatever. So they're going to go to the muscles, they're going to go to the glands, so you can have a motor output. Now, when we divide these up, again, some books will divide the afferent system into the visceral and the somatic. So the organ sensory versus the body sensory. Other books do not, because it's kind of difficult with that division, because some of the same neurons that pick up your body also pick up your organs. Again, this is why we have that referred pain that we talked about before. This is why your arm might hurt when you're having a heart attack, because it's the same neuron coming from an organ as coming from your skin. And you're going to perceive it as an arm problem because your brain's used to your body complaining more than your heart. So the afferent system is kind of harder to divide up as neatly because you have double agents. Okay, so sometimes it's the good guy, sometimes it's the bad guy kind of scenario. So it's not as easy as the division of the motor. Now, when we look at the motor division, when we look at moving the body, controlling the body, what are our two basic system types?
Say that again.
Basic system types for motor output. Think of it in the control scenario.
Antagonist? No.
So if we're controlling your body, what are two different ways, two different mechanisms for controlling the body?
It's autonomic.
Autonomic. So you have no control, involuntary. And then what's the other way you can control your body? If you got involuntary, you're also going to have voluntary. Okay, so that's our major division. Those you can control, tell them what to do, have input, and those you can't control. They're going to do what they're going to do whether or not you want them to. So the somatic nervous system and the autonomic nervous system. Somatic nervous system is going to the body, going to the skeletal muscles. Sometimes you'll see this called the somatic motor division because it's your body moving versus your organs moving. So this is your voluntary group of control. So it's going to the skeletal muscles so you can tell them to twiddle your thumbs if you want to twiddle your thumbs. It also goes to the skin because it's right next to the muscles. A lot of our automatic reactions like reflexes are going to go into this category because a lot of our reflexes are skeletal muscle reflexes to move us away from pain or to catch that ball or whatever other reflex we happen to be testing. The autonomic nervous division, the ANS, autonomic means autonomously, without control. You have no ability to control this. This is your involuntary group. You'll also see this called the visceral motor division because the organs fall underneath this category. So they're going to go to the things that are involuntary, like your heart muscle, like your smooth muscle, right? You can't stop your tummy from rumbling when it wants to, okay, because it's autonomic. And it's going to go to glands. You can't tell your sweat glands to stop sweating, right? Then we wouldn't need all the body sprays that are there pimping out for you, right? You guys stop it. Done. Okay, so that's not going to work for you. So glands and the hard and the smooth muscle. Now here we have a further subdivision because depending on what the body is experiencing, we're going to have different reactions. We have reactions when you're stressed and we have reactions when you're not stressed, right? You know, you have to forgive me. I don't normally act like that, but it was a bad day, that kind of scenario. So when you're stressed, that is the sympathetic division, your fight or flight responses. And you can remember that you sympathize if somebody's really stressed out. So your heart might be racing and your breathing might increase and your armpits might start sweating, all those things that we think happen when we're stressed out. And unfortunately, your body doesn't care what that stress happens to be. That stress could be a test today. That stress could be a lion trying to eat you. That stress could be, it's really hot in here. Your body's still going to respond exactly the same because it can't tell the difference between a test that's not going to kill you and a lion that will enjoy killing you. Okay, it's going to have the same kind of response, increased heart rate, sweating, all that kind of fun stuff. Then we have the sympathetic division, the I'm at home eating Cheetos division. Okay, I'm relaxed, I'm not stressed. Okay, so spring break, that's going to be the parasympathetic division for you guys. And it usually does the opposite of what happens when you're stressed, right? When you're stressed, your heart races. When you're relaxed, your heart slows down. Okay, so these two kind of counterbalance each other. So here we have our picture flow chart. And this one did do those divisions that I said that are optional. So they do the central versus peripheral. So central is the brain and spinal cord. The peripheral is the sensory division and the motor division. So the afferent nervous system and the efferent nervous system. They are alphabetical. Afferent goes towards, so that's your sensory. Efferent goes away. So A happens before E. So you got to go towards before you can go away from the spinal cord or brain. And you can remember the effector or efferent system goes to the effector organs. They both start with that EV. Our sensory, this one divided it between the visceral and the somatic. But again, this division isn't very clear cut because you'll have some neurons that do both jobs. They'll carry both sensory input from the muscles, but they'll also carry sensory input from organs, which makes it a little fuzzy on the line between the two. And then the motor division, not fuzzy. We have a distinctive division between our voluntary systems and involuntary systems. So our voluntaries, that's going to be the somatic motor division, the somatic nervous system, or the visceral motor division, the autonomic nervous system, going to the viscera versus the body. And that's divided into I'm stressed, sympathetic division, and I'm not stressed, the parasympathetic, because you cannot be stressed and relaxed at the same time. Just impossible to do. It's one or the other. This one is from your book, and they've just done the divisions based on chapters. Okay, so of course the nervous tissue chapter, this one we're looking at, this is giving us our foundation. So how do the cells function? What's the parts of the cells? Because again, the cells build to make the organs and so forth. So that's going to be chapter 12. Chapter 13 is the spinal cord and the spinal nerves. So that's all test 4 material. Test 5, we're going to have the cranial and brain. So chapter 14, we're going to have the SNS, the somatic nervous system, chapter 15, and then we're going to have the autonomic nervous system, chapter 16. So that's going to be your test 5. So this is a scary picture because it's the rest of your life. in AP1. Okay, so everything in one little nugget. Yeah. Worth way more than 1,000 words and some tears and stuff. All right. So when we look at the nervous system, we've learned that we have two types of cells. We have neurons and neuroglia. The neurons are the main guys doing the job. The neuroglia are your support. So then we're going to start with the main guys. We're going to start with the neurons. They are the structural and functional unit of the nervous system. Everything is built upon neurons, whether it's the brain or the spinal cord or the nerves or anything like that. And everything functions based on the neuron. So if your neuron ain't working, your brain ain't working, you ain't working. They have to be functioning at the cell level, which is why we start with the cells before we move on to the organ and how it works. Sometimes you'll see this sentence using the sciencey words, the basic anatomical and physiological unit of the nervous system. Because again, anatomical just means structural and physiological just means functional. Okay, so sometimes you'll see it the fancy way, sometimes you'll see it the plain way. Now, neurons have extreme longevity. They can live for over 100 years. They're as old as you are. Okay? And some of them are a little older because they were made during fetal development. So it's, your age plus like 3 months or 6 months, or depending on how old you are. Now, some of them are going to be younger. Okay? Not very much younger, but for the most part, a little bit younger, depending on which cells we're looking at. They have a very high metabolic rate, right? When someone stops breathing, what do paramedics worry about?
Oxygen.
Oxygen.
The brain cells, neurons.
Right, when you stop breathing, that's the first thing they worry about. Right? Oh, brain damage. Oh, they weren't breathing enough. No one goes, oh my gosh, save the kidneys. We want to make sure the kidneys are still working. No. Neurons require lots of oxygen, lots of nutrients. If they don't get it, they're little prima donnas. They just die. Kind of like those little princess people. If they don't get their way, they throw a fit, just leave. Okay, in this case, leaving means death. Okay, so we need lots of oxygen. We also need lots of sugar. Neurons only eat sugar. They do not eat fat. They do not eat protein. In general, the body uses sugar and fat for fuel, protein for building. So if anybody ever says, oh, I have a high protein diet so I can have plenty of energy, bless them, they don't know anything about science. They probably read something on the internet and they think it's right. Bless them. So much blessing. But when we look at neurons eat only sugar. They don't eat fat. They're going to use that as support. They're not going to eat protein. They're going to use that as support and build stuff. They have to have sugar to run. So this is why if it's been a while since a meal or since you've had some carbs, you might have a headache because you're hungry, because the neurons are in unison screaming, I want some freaking sugar, so they give you a headache. Or you might have, kind of fogginess if it's been a while since a meal, or if you're really stressed out, you crave sugar. Right? No one goes, oh my gosh, I've had such a crappy day. I really could use some broccoli. No, broccoli does actually have a lot of carbs. It's nasty to begin with. No, right? When we're stressed, we want a donut. We want a candy bar. We want a Snickers so we stop being hangry. That's why you get hangry. Those neurons are like, sugar, bitch, bring it. Okay? They want it. They got to have it. So they don't eat anything but the sugars. Now, neurons have no structures for cell division. When we look at cell division, when we look at mitosis, when you go through mitosis, your functioning has to stop. You can't do your job while you're dividing. It's kind of like when you're on maternity leave, you're not working, you're taking care of a baby, you're having that baby. So with mitosis, the cell has to stop, prepare for mitosis, go through mitosis, and then finish up. We don't want our brain to stop working because of cell division. It may seem like a lot of people's brains have stopped working, maybe for cell division, but it's not the case. And you can't survive with the brain stopping working. They have to keep going. Plus, these are really big cells. If you're 3 feet long and you divide in half, it's going to take hours, if not days, to do that. And you can't go without your brain working for days in order to keep going. So they don't have the ability to divide. They don't have centrioles or centromeres or asters or spindle fibers or anything that is necessary for division. In order to get new neurons, we have stem cells. They have to be built from scratch from these stem cells. Now, most of your stem cells stop reproducing, stop making neurons between 6 months and 4 years. So your spinal cord is the exact same size it was when you were 4. Your brain is a little bit bigger, but it stops growing at 5. Okay, so we grow into those funny structures. So for the most part, most of your neurons are just 4 years younger than your end date at the moment. Now, there are a few areas that still have active stem cells and production of neurons. One of those is the olfactory, the nose. And that's because your smell epithelium, olfactory epithelium, are actually exposed to the outside. Okay, they're sitting in contact with air. Because of that, they can be damaged or injured or killed if you smell the wrong thing. So that's why sometimes if you're exposed to a strong chemical smell, your sense of smell is off for a few days, you've wiped out some neurons and they've got to be replaced. And it may take a little while. Those cells can die in the case of a disease coming in. That's why COVID causes people to lose their sense of smell. The COVID virus killed the neurons that were smelling, and you have to rebuild them from scratch. And the older you are, the more time it takes for those stem cells to rebuild. So older people, it took longer for their sense of smell to come back once those stem cells rebuilt them. Yes, ma'am?
That's the same thing for like taste?
Yes, they're not as exposed as the nose is, and they're not going to reproduce as often, so those stem cells don't make them as quickly. Nose cells last 60 days before they die because they're just exposed to the front lines all the time. So they're very active in being able to be replaced by those stem cells. Your taste buds are a little bit more protected because they're tucked up and under your taste buds. So they're not exposed directly. So it's not like pixie sticks are going to wipe out your taste buds or anything. So since they're not as exposed, they do replace and repair using stem cells, but not as frequently. You also have some repair in the eye, but those ones are like really low down enough that we don't care that they exist. We also have these stem cell pockets that are very active in the hippocampus. That's your memory center. So you can always make new cells to store new memories. So that punchline for a lot of shows where it's like, oh, my brain is full, I can't learn anymore, or if I learn one more thing, I'll forget what my name is. It's not possible. Okay, so you can always make new cells for memories in that one area of the brain. The rest of the brain is pretty much done. It's not going to have any more cells that are going to be produced, but we still can produce some more here for memory. Yes, sir.
So you're saying the limit for memory on a human
Does not exist.
does not exist.
Unless you damage the hippocampus. If
Okay.
you kill the stem cells in new memories, which is a plot line for 51st dates.
Ah,
So we'll talk about that when we get to memories on test 5. Maybe we'll remember me saying something right now. Maybe not. Who knows? But yeah, as long as those areas are safe and protected and is deeper in the brain, so it's harder to damage those, you can still store those in memories. Look at that, it's a neuron. Ooh, pretend you care. Yes,
Ooh.
So it's like Alzheimer's and dementia in the hippocampus, that's damaged?
With Alzheimer's, the cells in the frontal lobe are dying at high rates, and they can't be replaced because there's no stem cells there. So you actually have massive cellular death in the front of your brain.
Is it only?
Yeah, that's how they can confirm you have Alzheimer's. They'll do PET scans and brain scans, and if your gyri are smaller, you've had massive cell death. So we had one. It was a guy in cadaver lab back in the day when we had cadaver lab, and he had Alzheimer's. That's not what killed him. And when they pulled the brain out, it looked like someone just shoved it in because his frontal lobe was almost gone because of the shrinkage that had happened. And that's why once you have damage from Alzheimer's, it can't be repaired because there are no stem cells there to replace those that have been killed.
That's right. They say it's almost incurable.
Are there any preventative measures you can take?
Basically, all you can do is slow things down. Now, if you catch it early enough, they do neurotransmitter therapies, and we'll get more into this when we talk about Alzheimer's with the brain chapter. But they can give you acetylcholine because that's what causes Alzheimer's is not enough acetylcholine, so you don't stimulate the neurons, so the neurons die from lack of stimulation. So if you give them more acetylcholine, you fully stimulate them, they're not going to die as quickly. But by the time you're diagnosed with early onset, you've already had cell death. So you can't completely reverse, you can just prevent progression and manage and slow down progression with that. So it's a managing disease, it's not a curable disease.
Is there anything you can do while you're still healthy? Just keep up with the acetylcholine?
Think. Yeah.
Think.
So
So brain games are,
yeah, they're exercise for your neurons because they have to send and receive neurotransmitters when you're processing stuff. That's not going to be a complete preventative because some people naturally get this. It could be hereditary, or as we age, we naturally decline in activity. But by staying active with your brain, just like by staying active with your muscles, you can try to retain some of that and lessen the loss.
Okay.
So there's our neuron. So the rest of the semester, he's the star. So next Thursday, when we get to the nervous tissue stuff in lab, we have a model that looks just like this. We'll be all up in this guy. So we're definitely going to know that little guy for test 4, for practical 4, for test 5. So knowing our guy and his parts. So we first start with dendrites. Again, dendra meaning branch. So these are branch-like extensions of the cytoplasm of the neuron. And the branches have branches just like tree branches have little branches that have sticks on them, or twigs or what have you. So these little branches off of the main branches are called dendritic spines. It's like you could call little twigs on a branch, little spines on a branch. They're going to make up 80 to 90% of your total neuronal surface. So that basically indicates that 80 to 90% of the function of the neuron is dedicated to the function of the dendrite. And the job of the dendrite is to receive information. They're going to have receptors. So they're going to have those little doorways that get opened or closed to neurotransmitters and bring information in. So this is how we can do the sensory input to that neuron. And of course, we have lots and lots of dendrites, so we can have lots and lots of sensory input. It's very similar to when you go buy a car, you don't walk onto the lot and point to the first one and go, that one, that's what I want. Right? Most people research. They look at gas mileage and price and insurance and blah, 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 colors. We look at a lot of stuff before we make one decision. That's because our neurons are built to receive a lot of stuff before we do one decision. So we act as an organism based on how those cells act, receiving a whole lot of input before we have an output. Now, dendrite length are going to vary. Some of your longest dendrites go from the tip of your toes, so you can feel if someone's tickling your toes, all the way up to the middle around your belly button to the spinal cord. So they can then drop off with the next cell coming in. So it's very common to have them upwards of 3 feet long associated with the extremities of the body. Now, in the brain, they might be much shorter because they're right next to each other. So they don't have to be that long, they can be micrometers in length. So depending on where they're going, where they're coming from, you'll have different sizes of parts of those neurons. Axons. We will have hundreds and hundreds of dendrites. We will only have one axon. Just like you have lots and lots of input before you buy one car. Okay, so we receive a whole lot of information before we send off one signal. And that's the job of an axon, is to send an impulse away from the cell body. Okay, so you can remember axons in a way, they both start with A. So they're sending information away from the cell body. Now, at the end of the axons, they're going to end with little branches. Okay, these branches, again, are called teledendria. Tele meaning far away, like television, telephone. Dendria meaning branch, so far away branches. We've also seen the terms axon terminal branch, that's cells. We can also call them terminal arborization, which just means tree-like structures at the end. So instead of ending in one single point, we have all these branches so we can communicate with multiple locations. Now, as we've talked about with skeletal muscle, when we take a normal cell structure and we modify it, we make it different, we give it a brand new name. So you're not just a line cook, you are now the line cook that's in charge of fish. And that's what you do, is you specialize in cooking the fish. So here we have some specialized terms. We have axoplasm. That is the cytoplasm, bless you, of an axon. Now, when a neuron is at rest, it has different ions in the cytoplasm than when it's firing. So at rest, it has lots and lots of potassium. When it fires, sodium's going to rush in, potassium's going to rush out. You're going to change the ion distribution during that activity. And because we're changing the ion distribution in the axon, we call it axoplasmic area or axoplasm. So the cytoplasm being different within the axon. And you'll have it with the change in concentrations of ions that will travel through. So different parts of the axon may have different components of the cytoplasm at any point in time. Now, this is also true of the dendrites. This is also true of the cell body. Unfortunately, a lot of stuff was discovered and named for the axoplasm side of things or the axon side of things. So they got all the special names. And by the time it went around to the other cell parts, like, yeah, nobody cares. But technically, dendrites. their cytoplasm changes if they're receiving or not receiving. And the cell body cytoplasm changes if we're firing or not firing, or if we're making neurotransmitters or not making neurotransmitters. So you'll have speciality across the cell fluid, but we only address it for the axons. We're a little axon biased because they're kind of a big deal because they're sending stuff. Okay. Axolemma, again, that is the cell membrane of the axon. The axon has specialized portions of their cell membrane along most of the length. It has a lot of extra sodium channels and potassium channels, so we can exchange, so we can have action potentials. By the time we get down to the telodendria, we have a lot of variation, so we can release neurotransmitters. So the telodendria cell membrane looks different than the axon itself cell membrane. It's also different than the cell membrane in the dendrite. The dendrite has a lot of receptors. So each section of the cell looks very, very different. It's just like your house. Your front door is on the front of your house. That makes it look different than the walls on the side of your house, right? They may just have windows, or maybe they have no windows. So different walls that make up your house have different functions, right? This one lets light in. This one lets south wind in. This one lets people out of the house. So same thing with that cell membrane. The axolemma is going to have a lot of structural changes to help it do its job. Now, your axons may have side branches so they can communicate with more than one cell or to different regions of cells. And those side branches are called collaterals or axon collaterals. And we learned with our ligaments, collateral just means side. Okay, so these are side branches of the axon. And of course, axons are going to vary in length. The ones that go to the tips of your toes so your toes can sweat, they're going to go from your belly button area down to your toes. So that's roughly around 3 feet. The ones that go to your toes that tell them to wiggle, they go from the top of your brain down to the middle of your spinal cord before they hand off to another cell to go to your muscles so you can tell them to wiggle. So very, very long. And of course, some of them can be short. So if they're just talking between two cells in the brain, they don't need to be very long to communicate with someone that's right next door. So there's that axon. And this picture calls it the terminal arborization, but it could have easily used the term telodendria or axon terminal branches that we saw when we looked at the skeletal muscle models. The collateral, again, popping out from the side so it can go somewhere else. Neurofibrils. So when you are a huge cell that's like 3 feet long, you need a lot of support, okay? Just like anything, right? If your kid is 3 feet long, they got a skeleton to support their body. These guys need a skeleton. That's the job of the neurofibrils. These are going to provide internal support so the cell doesn't collapse on itself, it doesn't shift or anything like that. Other than providing support, they can also spread nutrients. Okay? If we do the normal spread of nutrients, we just release sugar into the cytoplasm, it's going to drift and diffuse and get to where it wants to go eventually. Okay, it may take too long to get to where it needs to go. So if we put it on one of these neurofibrils, it's like putting it on a conveyor belt and just shipping it real fast. So it helps to speed up that spread of nutrients so we can get the sugars where they need to go. It also helps with depolarization waves, helping to spread action potentials. Nasal bodies. So these are our most active and well-developed rough endoplasmic reticulum in the body. So they're named after some guy named Niesel who discovered them. And again, we give them a special name because they're specialized. They can do more than the typical endoplasmic reticulum. So what do endoplasmic reticulum do in the average cell? Protein. They make proteins. All the proteins, whatever they happen to be. Now, in the case of the Niesel bodies, they're just not any proteins. They're neurotransmitters. So your neurotransmitters are protein-based. So this is why we need proteins. We need to build body parts, including neurotransmitters. Now, these guys qualify for a special name because they are so active. Whenever we tell our muscles to contract, for an example, if we want to pick up your bag of groceries and carry it in, we're going to have that neuron release acetylcholine, and it releases thousands of acetylcholine molecules just to initiate the muscle contraction. To keep you contracted, it has to keep releasing acetylcholine. So if you're carrying something for 5 minutes, you're releasing tons and tons, hundreds and thousands of acetylcholines every second. So you have to have tons of acetylcholine just to maintain muscle contractions. And these Niesel bodies are going to make all those from scratch. So this hiding.
So the nasal bodies make acetylcholine?
They make all neurotransmitters.
All, okay.
I'm just using acetylcholine because that's the one we've learned so far. Okay, so they have to make tons and tons more than the average. Not only that, when you look at an average cell, they might have 2 or 3 endoplasmic reticulum, enough to make the proteins to repair the cell or build a structure. In the case of neurons, they need so many neurotransmitters in such high quantities, 2 or 3 is not going to do it. They're going to often have way more than that, sometimes hundreds within the cell. There's so many nasal bodies, they give the color to gray matter. So gray matter is gray. Those parts of the brain, those parts of the spinal cord are gray in color because there's so many jam-packed nasal bodies that they're giving color to the cell. Right? It's the same reason why blood is red. Technically, plasma is yellow, and there's so many red blood cells packed in there, we see it as the color red. So we see these as the color gray because there's so many of them. So way more than the typical cell. Now, in your lab book, again, there's name changes that are going from naming them after a person to naming them after structures. It's slow going because there's so many things named after people, right? Just look at female junk, fallopian tube, Dr. Fallopian, Bartlett's glands, Dr. Bartlett's, all of the female stuff. Well, do doctors, okay? All over the place. So nasal, that one's easy. In your lab book, it calls it the polychromatophilic substance of the rough endoplasmic reticulum. You know, at some point, just call it nasal body. Who wants to write a paragraph to describe those little buggers? Okay? So I always use nasal body, but in your lab book, and we'll address it when we get there in lab, I'll have you cross it out because nasal is like 5 letters. It's easier to remember than polychromatophilic substance of the endoplasmic reticulum. Yeah, too many words. So there's our neurofibrils giving us support. There's our gazillion nasal bodies all over the place to make all those neurotransmitters so we can communicate efficiently with the cells around us. Myelin sheath. So the myelin sheath is found around the outside of the axon. It is a segmented whitish material because it's made out of fat. It's made out of phospholipids. And it's going to help act as an insulator, just like your fat helps to insulate you. It may not be very effective for us, but you know the reason why whales can live in the Arctic waters where it's freaking cold. They got layers of blubber, layers of fat to keep them warm. The thicker the myelin sheath, the faster the axon can function. It's just like when you look at a coat, the bigger the coat, the warmer the coat. So when you look at the myelin sheath, the thicker the myelin sheath, the faster the neuron's going to be, the quicker it can send off neurotransmitters, respond to neurotransmitters, so on and so forth. Now, with the myelin sheath, even though it does improve activity of the neuron, making it more efficient, the neuron doesn't have time to make it itself because it's so busy being a neuron. It's kind of like, I'm going to guesstimate nobody makes their winter coats. Now, so far in the 20 some odd years I've been teaching this, no one has said, I do. Okay? It's because you got to have special fabric and a special sewing machine and all that kind of stuff. It's better just to go to Walmart and buy it. A lot cheaper, a lot faster, and it's warm. Same thing here. Our neuroglia, our support cells, are going to make the myelin sheath, our coat for us, instead of the neuron. And because the peripheral nervous system has a different set of support cells than the central nervous system, we have different cells to do that. In the peripheral nervous system, they're called Schwann cells, named after some dude named Schwann. Their new name is neurolimal cells. Okay, so they're interchangeable on that one. In the central nervous system, they're called ogliodendrocytes. Isn't that a fun word? Oglio means few, dendro means branch, and cyte means cell. So it's a cell with a few branches. I like to picture them like partially shaved sea urchins, just because that picture in my mind is really fun. There's always a band-aid because there's an injury or something. Okay. So the areas that have the myelin sheath, they're known as the internodes. These are those big sections. So when you look at it, it kind of looks like a string of sausages. And ironically, almost any kind of cartoon that has butchers or pigs has a string of sausage somewhere in the background. You don't really see those often in real life, but they're on every cartoon. So you've got the sausage and the twisty, and then the sausage and the twisty. The sausage would be the internode, would be the myelin sheath. So here's showing you a Schwann cell making the myelin sheath. So it just kind of parks there, and it wraps around, and it makes it thicker and thicker and thicker. So the more you utilize this cell, the more speed it needs to have, the Schwann cell can add to it. Okay, so if you're using that neuron a whole lot, we can thicken up that myelin sheath so it can respond quicker. If you're not using that neuron a whole lot, the Schwann cell is not going to put an effort into making thick myelin sheaths, so the myelin sheath can actually get thinner. So they've done studies where they've shown if you maintain neural activity, you keep your white matter. You keep that myelin sheath so you can be fast. If you don't use it, you're going to lose it. Your body doesn't keep stuff just for the fun of it. It's like, if I'm not using it, it's dead to me. So if you're not using that neuron as frequently, the myelin sheath will get thinner. So you can change the myelinization to some degrees, not huge differences, but minor differences. Again, this is why you try to be active mentally, physically throughout your life to help maintain the activity of those Schwann cells and those neurons.
So neural repetition can lead to the degradation of this? Like just
Repeated
consistently
neuron use would lead to thicker myelin
doing
sheath.
it? Okay.
So if you sat there and didn't use it, it'd get thinner.
Okay.
The nodes of Rognier, it's French, or if you're from Texas, you can call it the nodes of Ranvier. It's kind of like if you shop at Home Depot versus Home Depot. Okay, crazy French people. Anybody else get the call yesterday when they canceled Dumas, but not us? The little person was like, the campuses at Dumas has been canceled. We're like, come on, now it's Dumas. It's not Dumas. It's not French. But if you listen to this, it is French because of the French doctor. So Rognier, or if you want to remember how to spell it, just call it Ranvier, whatever makes you happy. You're right, and you're not saying it out loud. So these are the nodes. This would be the twisty on the links of sausages or hot dogs. So this is the naked area of the axon. So there's no myelinization here. So this is where we're going to have a lot of sodium or potassium channels for exchange. This is also where the collaterals are going to pop out, because if you're a side branch, you don't want to dig through layers of fat to get out. You're going to pop out in an easy spot that's nice and naked. So as we look at the axon, you'll have interneuron nodes of Rognier, interneuron nodes of Rognier, interneuron all the way down the chain. The neurolemma. So we learned lemma meaning covering. So this is a thin membrane around the outside of axons. It is also called the sheath of Schwann, which sounds like a really crappy kid's music, like magician. Right? Oh, they've invited the sheath of Schwann cake or something. So with our sheath of Schwann, with our neurolemma, this is a thin living nucleated membrane. It's a little weird. It's made by Schwann cells. So these cells are building this living membrane that's around your axon to act as a little protector. That'd be like if your coat was alive and you had to feed it to keep it happy, but it would keep you protected from somebody touching you. I know, terrifying thought if your jacket attacked people. Okay, so this little guy will be found around axons in the peripheral nervous system if it's myelinated or if it's not. Okay? We don't have them in the brain and spinal cord because the brain and spinal cord do not have Schwann cells. This is the sole reason why your brain and spinal cord cannot repair. Okay, so brain damage is permanent because we don't have the neurolemma to help protect the developing new axon to regrow. They're exposed, so they're not going to be able to regenerate. They're not going to be able to survive. So permanent brain damage. In the body periphery, the neurolemma helps protect the developing and regrowing axon. It's just like if you want to have seedlings of plants, you put them in a greenhouse until they get big enough to be planted outside to deal with life. That greenhouse is going to protect them from the crazy weather. That's what the neurolemma does. It acts like a greenhouse. It is around the outside. It protects the axon when it has to be rebuilt. And it's going to ensure that it's safe and doesn't get exposed to anything on the outside. So without the neurolemma, you would not be able to repair neurons. So this is how your neurons can repair. It may take years, but they can repair in the periphery. The cell body, also called the soma, which is Latin for body, or pericheron, which is just fun to say. This is the biosynthetic center. This is where the nucleus is. This is where the organelles are doing the things cells do. So they aren't anything different than anybody else's main components of the cells. The Golgi apparatus, we learned Golgi apparatus packages materials to leave the cell. In this case, they're making synaptic vesicles. So they are also very well built and very active because they're packaging all the neurotransmitters to leave the cell. So we got to have lots of little zippy bags to put all those neurotransmitters in. They just don't get a special name because nobody apparently cared. So the plasma membrane, as I mentioned, depending on where we are, looks different. The plasma membrane, cell membrane of the dendrites would have a lot of receptors on it and some sodium potassium channels. The cell membrane of the parakaryon may have some receptors on it for neurotransmitters, but for the most part, it looks like a normal cell. And then the cell membrane of the axon, the axolemma, is going to have a lot of sodium potassium channels running along its length. And when we get to the very end, it's going to have lots of receptor drop off. Okay, so a lot of where we can release our neurotransmitters. So depending on where we are, each portion of that cell membrane looks slightly different because it has a slightly different function. So depending on where we are, we'll have those variations. So when it comes to functionality, what do we have to have the neuron work? So these are the three components that we have to have functioning. If they are not working, your neuron isn't working, you ain't working. So the first one are the dendrites. You have to be able to receive input. If you are in a completely dark room that you've never been in before, you really can't do anything because you don't know what's going on around you. You have no sensory input. So without sensory input, you can't have an output because you can't respond if you don't know what's there. So without dendrites, we're not going to be able to function. So we have to be able to receive information coming in. We have to have that conducting component. We have to have the axon traveling away from the cell body to the end so we can communicate with the end so we can release. And then we have to have the secretory output, the axon terminal branches that actually release the neurotransmitter. Okay? So if we don't release neurotransmitter, we keep all of our secrets. We don't tell what's going on. So these areas are the areas we worry about. If you damage your axon, we're deeply concerned. If you damage a dendrite, we're deeply concerned because it can alter the function of that cell, leading to the death of that cell. Also, when we look at medications, this is where medications target. If we want to block something, we may go to the dendrite and say, don't receive this neurotransmitter. Done. If we want to increase a response, we might go to the axon terminal branch and say, release a whole lot more. Okay, so some of your muscle relaxants, if you've got twitchy muscles, that drug will go to the axon terminal branches and block the release of acetylcholine. No acetylcholine, no initial trigger for muscle contraction, your muscle stays relaxed. So that drug is going to go to the axon terminal branches to prevent release. Some drugs, again, go to the dendrites. If we want to make you happy, we can give you a medication that opens up more receptors or make your receptors more sensitive to a neurotransmitter so you can be happy. So they might target the dendrites to help those things open up a little bit more efficiently or shut them down so they can't receive. So if we have a drug that blocks the dendrites, you're not going to receive information. So if you need to go to bed and you need to get some sleep and you just can't turn your brain off, you can take a pill that blocks the dendrite. Your brain can't think anymore because it can't receive input. So it's going to help you to fall asleep. So a lot of our medications are going to target these areas because it targets the ability of that neuron to function properly. Yes, ma'am.
I'm not sure if this goes exactly like this, but if you have like an itch on your body and you scratch it, how come sometimes you'll feel it in a completely different part of your body, like the inside?
Those sensory neurons, some of them have a wide field that they're monitoring. So it's kind of like if you're outside and you hear somebody yell in the park, but you don't know where in the park they are, so you just, I guess them that way. Your body does the same thing. They've got their own realms that they monitor. And if you touch one part and it can't tell because they have such a huge field, you might feel it as coming from somewhere else.
So if you're scratching, you can't find that place, that's what that sensor is.
It could be, or when you're scratching, an itch is a precursor to pain. So something is annoying your neurons enough for it to be registering, but not enough for it to hurt. So if you're not finding that spot that's receiving and removing whatever it is that's stimulating that area, the itch lingers. So it doesn't go away properly, especially if it's an internal, because then it could just be neurotransmitter trickles just enough to annoy yourselves. And there's nothing you can do but curse them. Right, nuclei versus ganglia. Again, lots of terminology. A nuclei is a cluster of cell bodies in the central nervous system. They kind of hang out together, just like students like to hang out together. So when you go to the bookstore and you see a bunch of people at a table talking, or go to the, the study center, and they're all talking at it. That would be a nuclei. Nuclei in the brain, we have things like the red nuclei, the basal nuclei. So we'll be using this term a lot, especially in the brain, but we will also in the spinal cord. Ganglia, cluster of cell bodies outside. So instead of hanging out in the coffee shop here or the study center here, you and your friends go hang out at Barnes and Nobles. You're still clustering together, but you're not on campus. So you're not in the hub of Amarillo College, you're outside the hub. So they're the same thing. Both of them are clusters of nerve bodies or neuron cell bodies. It's just who's in the central nervous system, who's out of the central nervous system. So location different. A synapse. So when we have a synapse, this is a functional connection between a neuron and another cell. So this might be between a neuron and another neuron. It might be between a neuron and a muscle, that kind of stuff. Synapse. So when we have our synapse, in order to communicate, we have to have a neurotransmitter, right? When you call somebody, you have to have something to communicate with them. Right? Very few of us just call to hear them breathe. Creepy. Right? You're going to talk to them. You're going to have a message. So examples of some neurotransmitters, acetylcholine. We've already talked a lot about that, communicating with skeletal muscles. Norepinephrine, serotonin, dopamine. We'll be getting into a bunch of different types of neurotransmitters as we progress. Now, if a neurotransmitter substance remains in contact at the postsynaptic membrane, that stimulation, that impulse will continue indefinitely, right? If you're talking to somebody and they won't shut up, you can't get off the phone, right? And then it's like, oh, 2 hours in, okay? In order to get that person off the phone, you have to do something, right? Oh, my batteries are going dead. I've got to let you go. Or somebody's at the door. Whatever excuse you want to use, okay? You got to have something that says, shut up. In the case of neurons and neurotransmitters, that's an enzyme. In order to stop acetylcholine, you got to have something to break down acetylcholinesterase. So without that enzyme, the communication would continue indefinitely, right? Without telling the person to shut up, they'd keep talking forever. Okay? You've got to have that stop point. So here's our lovely synapse. So here's our pre-guy, our post-guy, and they're communicating. There's those little synaptic vesicles releasing the neurotransmitter so we can stimulate that neuron. So, as mentioned, there's a lot of definitions, a lot of terminologies. In nervous tissue, you just don't go, that guy sends, that guy receives. That guy has a special name, and the other guy has a special name. We just have to learn all the special names. And that's half the battle with the nervous tissue, is learning the terms and then applying the functions of those terms. Okay? So we start with the presynaptic neuron. The job of the presynaptic neuron is to send information to the synapse, hence the word presynaptic neuron. Right? It's the one before, like the pre-game is before the game itself. Now, in order for that presynaptic neuron to release, it has to have a special membrane called the presynaptic membrane. So it has a special way to release those neurotransmitters, those little messages. And that message is going to float across space, which is the synapse, and get to the other guy, the postsynaptic cell. So the postsynaptic cell is going to receive the information coming in. And again, that's why it's called the postsynaptic cell. It's the guy after the synapse, just like the postgame is after the game, or your postscript, PS, that's after the letter, in case you ever wonder what PS stands for. Remember writing letters back in the day? PS, more information. Postscript, okay, after the main letter. So they're going to be receiving our information. And in order to receive the information, they have to have special membrane to do that. They've got the postsynaptic membrane to allow for that information to come in. Okay, so we've got specialization here. Now, when we look at the parts of our neuron, what parts of our neuron send information? Who's going to be the sender in a neuron? The axon. Axon sends. That's its job. So when we talk about the presynaptic component, usually we're talking about the activity at the axon. Who or what part of the neuron is going to receive?
Terminal branch. Dendrites.
Dendrites. So our dendrites receive. So if I have a drug that blocks the postsynaptic membrane, what part of the cell is it targeting? I get that a lot. If I have a drug that blocks the postsynaptic membrane, what part of this cell will it target?
The dendrite?
The dendrite. So if I have a drug that blocks this, that's going to affect my dendrite activity. Does nothing for my axon. It's still functioning. If I have a drug that blocks presynaptic activity, then it blocks this guy. Targets the axon only and not the dendrite. And again, this is one of the reasons why we have so many different types of depression medications. Some of them target this, some of it targets this, some of them gives you a whole bunch more of those, increasing your chances to get it. We have different response times. So these terms are going to be important because, again, we just can't say the sending guide, the receiving guide. You got to use their titles. And when you look at the paperwork that comes with your drug medications, it may say this is going to affect the postsynaptic membrane of the receiving cell. Dendrite stops you from receiving a neurotransmitter. You're still sending them, you just can't receive them. And that's going to let you relax, or whatever that drug is going to do. Or diseases, they're going to affect different things. So we talked about myasthenia gravis on test 3, right? That's that autoimmune disease that attacks the neuromuscular junction. It specifically attacks axons. It attacks the presynaptic membrane. And if the presynaptic membrane can't send acetylcholine, the muscle can't contract. So it causes muscle weakness. So it's going to target this little guy. Now, if we want to use analogies, we can use sports stuff, right? Pitchers. What's the job of the pitcher?
Pitch the ball.
Pitch the ball. Throw the ball. Be like this guy here. He'd be the presynaptic guy. He's going to throw the ball. Who does he throw the ball to?
The batter.
Is he throwing it to
umpire?
the batter, or is he trying to get it to the
Umpire. No.
catcher?
Catcher. Umpire.
You can hit the umpire if you want to. They kind of frown on that, though. Okay? Right? When the pitcher throws the ball, he's pitching to the catcher so he can catch the ball. Because if he catches it, that means the batter missed. So that would be your postsynaptic cell. Now, does your batter and your catcher have the exact same glove?
No.
No, right? Catchers have a catcher's mitt. It's made extra padding to receive the ball. That would be the postsynaptic membrane. It's built different. It has those little receptors so it can pick up that neurotransmitter coming in. The pitcher has a different glove. Okay? It's going to help, with the throw. Pretend it helps with the throw. It looks very different, acts very different. It sends the neurotransmitter. Now, if you throw a football at a catcher, can he catch the football?
No.
No, it's the wrong shape. Okay, it's not going to fit in his catcher's mitt. So if we throw in something like norepinephrine into this receptor here, into the synapse here, it can't attach to the receptor because it's the wrong shape. This is, again, why depression medication doesn't always make you twitch, because muscles have acetylcholine as their neurotransmitter, skeletal muscles, and our depression drug might be norepinephrine, completely different neurotransmitter, so they're not going to affect each other for most parts. Now, if you were to throw a tennis ball at a catcher, could he catch it with his catcher's mitt?
Yes.
Yeah, it's close enough. You can throw a baseball, you can throw a softball, you can throw a tennis ball, if you skilled, a ping pong ball. They're the right shape. They're not exactly the same, but it's close enough. This is how some of our drugs work. So if you have nicotine, okay, because you're taking your outside source of nicotine would be like throwing a tennis ball at this catcher. It's the right shape, not really acetylcholine, but close enough that this guy will catch it and we're going to respond to it. And so some of your drugs, that's how they're able to work. They're close enough to that receptor, for that receptor to receive it. So when we're talking about these are the terms we're going to use. Okay? So the dendrites are always our receiver, so they're going to be associated with the postsynaptic cell, the postsynaptic membrane. Our axons are always the sender, so they're going to be associated with the presynaptic cell. They're going to be associated with the presynaptic membrane.
Did you choose nicotine because it's a stimulant?
I choose nicotine because it does mimic acetylcholine.
Okay.
That's the sole reason why it gives you all the things nicotine gives you. And we'll actually talk about nicotine versus muscarinic poison or shrooms when we get to test 5, because they are very close mimics to acetylcholine. All right, so there's our players. So our pre-cell and our post-cell. So we can see here's the axon, and he's going to talk to the dendrites. And this one does actually tell you the axodendritic synapse between the axon and the dendrite. Now, most neurons function both ways, right? Catchers can throw. Pitchers can catch. But they're going to do it slightly different. They're not going to use the same glove. Okay? So this guy who is the presynaptic cell in this communication, he can still be a postsynaptic cell, but his dendrites on the other end are doing the postsynaptic activity. So the axon is never going to receive, but if this guy wants to receive something, it'll engage a dendrite. Okay? So our catcher isn't going to use the same glove to catch as he is going to throw. Right? He'll use his hand to throw instead of a glove. Now in the body periphery, the receiving, the postsynaptic cell, isn't always a neuron, right? Sometimes the pitcher throws the ball not to the catcher, but to the guy in outfield, okay? Sometimes he might throw the ball to a fan because, I don't know, why not, okay? They're going to be somebody else. They're a neuron talking to a muscle. We have a neuromuscular junction, which is what we talk about a lot for test 3 and practical 2, that neuron and muscle hookup. The neuroglandular junction is when the neuron is talking to a gland. So if it's time to sweat, the neuron is going to release acetylcholine. That will then tell your sweat glands to release sweat so you can be sweaty. So neuroglandular junction. When a neuron talks to another neuron, we don't call it a neuro junction because then it sounds like your record is skipping. We call it a synaptic knob. So when we see synaptic knob, it just means 2 neurons talking to each other. So this would be a synaptic knob because I made them both neurons. Axoplasmic transport. So with this, it was first discovered and described using axons and their activity. After doing that, when they studied more parts of the cell, they realized, well, dendrites also do this. And some cell bodies also do this. So instead of saying axoplasmic transport for just the axons and dendroplasmic transport for just dendrites, they call it all axoplasmic transport, but it doesn't get isolated to the axon. Okay? So it's a misname. Any part can do it. So this is just movement of material, like ions, between the cell body and the synapse. Okay? So if we go towards the synapse, that is known as anterior grade flow. So if we see a cell doing anterior grade flow, we would suspect that's the presynaptic neuron doing that. And we would suggest that that's the axon that is firing. Retrograde flow. Retro means past, so going back to the cell body. So they're going away from the synapse towards the cell body. So if we were to see retrograde flow in normal cells, we would expect that this is the postsynaptic cell that is responding. We would expect this is the dendrite. So if you have a drug that prevents retrograde flow in a neuron, you know that that drug prevents the postsynaptic cell from receiving. That drug is going to affect the dendrites, and you're not going to be able to receive a neurotransmitter. So again, these are those big words. They sound really good when you put them together in a sentence. We just have to translate them down to catchers and pitchers. Okay. Now, not only can we use this to identify cells and normal cell functioning, we can use this to help identify how drugs work. So if you put this drug in a petri dish with the cells, and we notice all the cells are doing retrograde flow, we know that drug is stimulating dendrites, and we're increasing the activity of the postsynaptic cell so they can see how it's working. They may not understand how it's working, but they can see what cell is receiving and how it's going to maybe respond, make it a better place with that drug. We can also use it when it comes to studying diseases. So one of the big ones they study is rabies. So when you have rabies, now rabies does not look like this. I did not take micro, so I do not know how to draw proper rabies viruses. This is probably a bacterial phage. Ooh, someone's excited about rabies. Okay. So what happens when you're bitten by a rabid animal? The rabies virus uses retrograde flow. So when you're bitten, it's going to go to any dendrite it can find, or excuse me, any cell it can find. And it's going to use retrograde flow. Go, rabies, go. to go to the cell body. Once they're in the cell body, they're going to do what all viruses do, replicate and replicate and replicate until they blow up the cell, releasing rabies for everybody. So it kills the cell. And magically, neurons talk to other neurons. So if you have rabies and you blow up, your rabies splatters over the next cell, it sucks in using retrograde flow, and it goes to the next cell, and the next cell, and the next cell. So that's how they can predict how it spreads through the body. If we can stop it, we stop the progression of the disease. Now, unfortunately, with rabies, it is 99.999999999% fatal. And that's because 2 people have survived it successfully. Everybody else are in some stage of medically induced coma until they die.
Wow.
So once it gets to the brain and spinal cord, when it kills those cells, you can't replace those brain and spinal cord cells. So if you lose enough cells in your brain, you die. So that's what happens when you get the rabies virus. Now, if you are bitten by an animal and they want to keep you alive, they'll give you antiviral medication, and they give you lots of shots to your abdomen. And that's to basically line up around the spinal cord, acting as that final barrier. So as that rabies is coming in using retrograde flow, they hit that antivirus and antibody issues, and it can fight off rabies before it gets to the brain and spinal cord. If it reaches brain and spinal cord, you've got 2 weeks before you die. It will be fatal. The ones who have survived, the first one was an athlete in high school. She was bitten by a bat. Don't pick up bats. If you can pick them up, they're sick. Right? But they put her in a medical coma for a year, bombarding her brain with antiviral medication. When they woke her up and she survived, it basically wiped her clean. They had to teach her how to eat again, how to walk. She was like an infant at 19. And they had to start her all over again. But she was able to do that because her brain was young enough it could bounce back. The other kid was 9. Most of us not going to bounce back. And who can afford a medical coma? Those are expensive. Okay, so you're going to die if you get rabies. That's also, in theory, the basis of our werewolf myths. Because if you're bitten by a rabid wolf, well, it's a wolf not afraid of people, and within a fortnight you're howling